next speaker here is my own student, Wengong, and he will present work that he did on drug combinations. So Wengong.
Hello, everyone. My name is Wengong. Today I will talk about my recent work on developing predictive models for searching COVID-19 drug combinations. So drug combination has been developed for many different diseases. And for example, in HIV, HIV therapies are primarily now centered around multiple drug combinations involving multiple drugs, targeting different with different mechanism of actions. So our goal is to build models to predict effective drug combinations for SARS-CoV-2. And recently, NCAT has released 2 different screens for drug combinations with labeled drug combination synergy data. It has around 160 drug pairs in total. So the challenge here is how to model, how to learn to predict the synergism of drug combinations with limited training data. So graph convolutional networks are powerful tools for drug discovery. Now it has been de facto models for molecular property prediction. For example, if we want to predict the antiviral activity of the compounds, you can just train these graph convolutional models to learn these disease-specific fingerprints to predict the antiviral activity. However, the graph convolution models are very data hungry. It will have a poor generalization if you only train it on, for example, these 160 drug combinations, right? So this applies to every model, every deep learning model, and graph convolution is no exception. But there are some sort of inductive biases that we can leverage here to improve the model generalization under this data limited regime. So we know that drugs are, they typically interact at the biological level. For example, the crucial information would be like what's the mechanism of action for these drugs, like what biological targets it binds or what pathways that it interacts with. So we want to add these additional information to the model so that it can learn how to reason about biology or what biological targets that these drugs are interacting with. So the question here is what makes a drug combination effective, right? If you think about it, or if you look at these HIV antiviral drug combinations, you will see that first, each drug in the combination should be potent, right? Each individual drug should be able to deliver some level of antiviral effect, even though it may not be strong enough, but at least there should be some signal. And this motivates us to sort of incorporate the single agent antiviral screen in the model training. And I will talk about later how this would be integrated with the drug combination synergy prediction. But also more importantly is sort of this intuition that synergistic drug combinations may have like complementary mechanism of actions. For example, like some of the HIV drug combinations, one of them target, say, the viral protease, and the other drug in the combination targets, say, the viral entry protein. For example, in that case, will be CCR4 or I forgot the other name of the drug of the target, but basically try to inhibit the target that the virus leverages to enter the cell. So this motivates us to incorporate this additional biological information of drug target interaction. So we want to model what targets that each drug interacts with. And there are two types of targets. One is the viral proteins like viral protease, or in this case will be, for example, SARS-CoV-2, the 3CL protease, or we have the host protein that the virus leverages to enter the cell. Like in this case will be ACE2, and we want to find drugs that inhibit ACE2. And also recently there's a well-received paper from UCSF, which identified around 300 targets that interacts with the SARS-CoV-2 protein. So we want to incorporate this information to the model when we reason about drug combination synergy. So to be more specific, we propose a simple yet effective approach by decomposing the graph convolutional model into two components. So the first part of the model it's responsible for learning drug target interaction. And the output of this drug target interaction module would be we call drug target interaction vector. It will be a list of binary variables. Each represents whether this drug inhibits or activates, whether it inhibits these biological targets. For example, here the first entry could represent the 3CL protease, or the second one could be representing the ACE2, and so on. So in total, we have 2 viral protease and 1 viral entry target, and also around 33 host proteins that identified by this Gordon identified by UCSF paper, and we incorporate them into the drug target interaction module. And the second module is this target disease association module. Basically, it assigns the importance of each biological target with respect to the SARS-CoV-2 antiviral activity. Basically, if this gets a high weight, that means inhibiting this will be very important for this antiviral activity. And of course, the original UCSF paper identified much more biological targets. They have around 335 host proteins, but unfortunately, we only have 33 of them that have drug target interaction data in, for example, Campbell database. So this means that given that there are a lot of information that will be missing in this drug target interaction vector. And this will definitely limit the model's performance. So we propose a simple technique by extending this drug target interaction vector with additional latent targets to account for this missing biological information. So basically, these latent targets are free parameters learned by the model that is adjusted through the single agent antiviral activity assay, antiviral activity data. These are, at this point, these will not carry specific, will not correspond to any specific biological targets, but we will show it later that these latent targets are found to be associated with some biological targets that are actually relevant for SARS-CoV-2. So to train the model, we will leverage, we will train each of the model jointly through 3 objectives. The first one is this drug target interaction loss. So in order for in order for each neurons in this vector to correspond to each biological target, we will train these neurons to be predictive of, say, the drug target interaction labels. For example, we will, so here we feed in, we train the model on this of this data of molecules and whether it binds or inhibits each of the biological targets. And this can be just a simple cross entropy loss. These data are collected from various sources. For the viral protease, it is collected from UNCATS, and for others are collected from a Campbell database. The second module we need to train is the target disease association module, right? So here to sort of assign the importance weight of each biological target, we train the model on the a single agent antiviral assay. So this is from UNCAT, which contains around 8,000 compounds with their labeled activity against SARS-CoV-2. So the whole model is trained on the single agent activity data, which allows us to fine-tune these latent targets so that it will carry relevant information rather than just totally being free. Now, lastly, we have this drug combination loss, which is the center of the model. This is the main goal of our approach, right? To predict what's the synergistic effect of the given drug combination. So here, the input to the model, now instead of being a single drug, now we have two drugs, and in principle, it can be extended to multiple drugs as well. So just for simplicity, let's look at here just two drugs as input, and we feed them to the drug target interaction module, which will give you two vectors, each representing what are the targets that this drug interacts with. So now if you look at this drug target interaction vector for each drug, we need to aggregate them in order to capture how they, what's the aggregated effect over this set of biological targets. So we adopt this famous Bliss independence assumption by taking the sum of so basically what we do is we take the sum of these two vectors and minus by subtracted by the multiplication of these two vectors. So basically this assumes that if one drug interacts with this target, then the combination will be interacting with this target. So once we have this aggregated drug target interaction vector, we then feed it into the model, the target disease association module, to predict the combination effect, C12. And this represents how effective it is, this drug combination. But in order to quantify synergy, we need to compare it to the additive effect of these two drugs. So to do this, we feed these two drugs individually to the model to predict its respective drug antiviral activity. So here we have S1 and S2. These are the predicted antiviral activity score for each individual drug. And then we can compute, again, by utilizing this Bliss independence assumption, we compute the expected effect of these two drugs. And we then quantify the synergistic effect of this drug combination by taking a difference between C12 and S12. So the main difference between these two quantities is this aggregated vector, right? So when you feed these two drugs individually, there is no interaction in terms of the biological targets. While when we predict the combination effect, we have this drug target, we have this aggregation of the two drug target interaction vectors. So we have a quick a summary of our results. So our training data is comprised of three types of data. First is the drug combination assay, and the second one is the single agent activity assay, both from UNCATS, and we also have drug target interaction data, around 20,000 of them compiled from Campbell. So the validation set is a tiny set that conducted by this Nature paper from the reframe group. Basically, they have around 20 combinations, which all involved remdesivir. And the test set is comprised of this drug combination screen from UNC. They have around 70 drug combinations with labeled synergy or antagonism. So we actually have more results of different ablation studies of the model, but just for simplicity, let's just compare our model with respect to standard graph convolution. As you can see here under fivefold, so basically we run our model 5 times with different random seeds, and as you can see here on the right-hand side, the model by modeling drug target interaction, we can actually does a lot better than the standard graph convolution that is trained only on the drug combination plus the single agent assay. So basically, this model is trained by removing all the drug target interaction, and this will be just purely reasoning over the latent targets. And as you can see, the standard deviation is also a lot smaller compared to the standard graph convolutional models. Lastly, I just want to quickly describe what does the latent target actually carries biological meanings. So here we did some simple analysis of whether these latent targets actually corresponds to any real biological targets in human body. So we found that this is after the fact. We found from this Riva et al.'s paper that they actually identified around 50 different biological targets that they think is related to SARS-CoV-2. So what we did is for each of the latent targets, we try to see whether it is predictive of the drug target interaction for each of the biological targets identified by this ReframeDB paper. So we can just simply measure the AUC of each latent target if you have a list of drugs and whether it binds to this real biological target or not. So from the right-hand side, you can see that these are the list of latent targets, and each edge means it has AUC over like 0.7 with respect to the real biological targets here. And you can see that there are quite a significant association between these latent targets and the biological targets here. And we think these latent targets are capable of identifying even some new targets that may be relevant for SARS-CoV-2, which are not still yet to be identified. So just to conclude, I presented a model that's for predicting drug combination synergy in case for SARS-CoV-2. The same methodology can be applied to cancer as well, as we are collaborating with UNCAT on the pancreatic cancer drug combination, which basically you can adopt the same methodology. And we developed a model that builds on various biological information. And the key takeaway is bringing biological information is crucial for a modeling drug combination. And also to account for missing biology, we need to allow the model to learn these latent targets, the free parameters, through the single agent activity data. And so with that, I hope I'm on time. I will take some more questions if there's more time.
So thank you very much, Wengong. So there is a whole bunch of questions. I would let you select 2 questions that you want to answer. The rest you can answer over text. You just need to be very brief. So select the 2 questions which would be fast to answer.
So I saw one question is how to reduce the data noise and false positive data from disease and
No. Sorry, your question start here, so you can let me just
okay, maybe
select the question. The first one I would just select from Pat Walters, who was featured in my presentation.
Okay.
How are activity thresholds defined for DTI vector? Are activators treated differently from inhibitors? Where only high confidence value from Campbell used? Those were 3 questions, actually. Do you see it?
That's a lot of questions to process. So we are for the drug target interaction, the threshold is actually determined by Campbell database itself. So there is a paper which they released 2 years ago. They basically determine the thresholds based on the different protein families. For kinase, it will be around, I remember, it's like 30 nanomolars, and for others, it could be just 1 micromolar, for example. And we just collected the process. So we didn't do processing or binarization by ourselves. We just collected the binarized data that collected by processed by the Campbell EBI group. And for what's the second question?
The second question, gosh, where did it go? I just saw it and it disappeared.
I remember it's also related to like high confidence.
No, I see.
Again,
I see.
We removed all the targets that only have, say, around 10 data points, because we believe that if you train the model on the biological target with less than 10 data points, it will be very noisy. So we did that thresholding as well, just to reduce the noise in the model training.
So great, Wengong, I think you should just go and answer all the questions. Thank you very much for the great talk. And now